Welcome to the lecture on bacteria and viruses. This is chapter 19 in your book. The first thing I want to do is go over some of the objectives I'm going to be covering in this video, and I'll come back to it in each section. The first section is on bacteria. I want you to be able to distinguish between eubacteria and archaeobacteria. I want you to identify what characteristics scientists use to classify prokaryotes or bacteria. And I want you to describe the process by which bacteria reproduce. Okay, so the first thing is we are going to use these words, bacteria and prokaryotes, kind of interchangeably. If you'll recall, prokaryotes have no nucleus. Okay, so make sure you write that down if you don't remember. In the case of bacteria, so I'm going to draw bacteria here. In the case of bacteria, their DNA is this circular structure found inside, so our DNA is found in chromosomes. They have like a circular DNA in bacteria, but they have no nucleus. Okay, so there are two kingdoms of bacteria. You may recall this from when we were learning about classification and Carolus Linnaeus. Eubacteria is the one that's much more common in the world. It's the things, most of our diseases are caused by eubacteria. This particular bacteria here, this figure is in your book. This is an E. coli. So let's take a look at this. They have a cell wall. They have a cell membrane. They have ribosomes, which, as you know, are used to make proteins. They take pieces of their circular DNA, this is their DNA, and they make proteins out of them. And so they're going to need those ribosomes just like you and I, but they don't really have any other organelles. That's pretty much it. Their cell walls contain a chemical called peptidoglycan. Archaeobacteria are different though in that they lack that peptidoglycan in their cell walls. So that's one difference between them. This right here is called a flagellum. Many bacteria have things like this in order to move from place to place. E. coli is one of those that does. And the final structure here on our E. coli is what's called pili. And it's used to form structures in between two different bacteria. We're going to talk about that when we start talking about the reproduction of bacteria. That's where that is important. So let's move on to archaeobacteria. Of course, archaeobacteria lack peptidoglycan. They have different membrane lipids and genes. So actually what's interesting is that some of the genes that you find in archaeobacteria can be found in eukaryotes like you and I, and plants and fungi. So they're genes that you don't find in eubacteria, but that you find in archaeobacteria. So for that reason, scientists consider archaeobacteria an ancient ancestor of everything. Now, whether that's true or not, I don't know, but that's why they're called archaeobacteria, because they're considered ancient. And then, of course, unlike eubacteria, archaeobacteria live in really harsh environments, extreme environments, you might say. You can find them in the bottom of, glaciers. You can find them in hot springs. So they're really interesting and very different from eubacteria, and that's why they were put into two different kingdoms. All right, there are a couple of different ways that scientists classify prokaryotes. And the first one is they just simply look under a microscope and they look at the shape. And there are three basic shapes to bacteria. There is the spherical shape, and scientists have called this cocci or coccus. There is a spiral corkscrew-shaped bacteria called spirilla. And finally, there are rod-shaped bacteria called bacilli. So make sure you write those down in your notes. Another way that scientists classify prokaryotes is they will look at the cell walls of the different prokaryotes, and they will look for different chemicals that are in the cell walls and how they react to other chemicals. So they do a stain, it's called a Gram stain. All you need to know basically is that they look at the chemicals in cell walls to classify bacteria or prokaryotes. They look at movement. Not all bacteria are able to move, but some of them have flagella or cilia or something like that that allows them to move around. And finally, they look at how they obtain energy. So if you'll recall, again, from when we looked at classification, some bacteria are heterotrophs, which mean they have to eat other things. Maybe they use chemicals or chemoheterotrophs. And then some of them are called autotrophs, so they make their own energy. So in this case, they have to take in something in order to get energy. And in this, they use chemicals or they use light in order to make their own energy. Okay, now let's look at reproduction of bacteria. There's two main ones that we're going to look at. The first one is binary fission. And in this one, this bacteria forms two bacteria, okay? And basically just splits apart. So it's kind of like mitosis. Oops, I'm going to make the DNA the same color here. So this is, when you think of like mitosis, this is sort of the same thing. Binary fission. Bi means two. Fission means to split. So this is essentially where the bacteria grows and splits into two bacteria. Conjugation is interesting because this is where that pili that I was talking about before comes in handy. These little, it forms like a bridge between the two bacteria, this bridge, and then they exchange genetic material. So they can swap genes and exchange genetic material. It's pretty cool. And then finally, there's something called endospores. If there's harsh environments that the bacteria is exposed to, then sometimes they can form what's called an endospore, and the bacteria kind of dies, but the spore stays around. And then when the environment becomes more conducive to growth and metabolism, then the bacteria will come back to life, kind of. It's interesting. All right, so I think that's it for bacteria. So as you can see, we distinguished between eubacteria and archaeobacteria. We identified what characteristics scientists use to classify bacteria, and we talked about how bacteria reproduce. Okay, now let's look at viruses. Now, virus structure is very simple. There is DNA or RNA, and there's a protein coat called a capsid. Now, what that protein coat looks like can be very different. I've put a picture here from your book. This is a bacteriophage. It looks kind of like a spider. This is an actual picture of this thing. It's so weird. This is the tobacco mosaic virus. Again, this protein coat looks very different. And then this should look familiar because it does look like our coronavirus that's going around right now. But this is a typical influenza virus. And so you can see, just because it has only a couple of things, it can attack a lot of different types of cells, and it can look very different. There are two basic kinds of viral infections using DNA. So a virus, as I said before, can have DNA or it can have RNA. So we're going to talk about viral infections using DNA. Now, an important thing to note here is that viruses are not considered living. And the reason that is because of these two different basic kinds of infection. Viruses cannot, on their own, metabolize energy, grow, and reproduce. They need a cell to reproduce for them. If there's no cell reproducing the virus, then the virus doesn't reproduce. It cannot reproduce all by itself. So it's not considered living by scientists, which is pretty cool. Okay, so there's two different kinds of infections. In a lytic infection, the DNA enters the cell and basically takes over, okay? And it starts to use the mechanisms within the cell. So it uses the ribosomes. It uses all the things that our cell is doing to make our own proteins and to function. And this thing comes in and it hijacks all of that. And it just starts to make copies of itself. And it actually destroys the DNA in the cell, of the cell itself. So it kills the cell, and it just basically becomes a virus-making machine until eventually the cell bursts and there's a bunch of viruses now that have been created by the cell. A lysogenic type, so that's a lytic infection. You can think of like to lyse something means to split it. So a lytic infection takes over the cell and then splits it apart. A lysogenic infection is a little bit more sneaky. And this is where the DNA actually becomes part of the cell's DNA. So this is where the DNA is injected. The DNA becomes inserted into the cell's own DNA, and it kind of waits, it kind of waits for the right opportunity or the right time, and then it turns on and it starts making copies of itself, and it becomes lytic. Okay. And finally, we have what's called retroviruses, and their genetic material is made of RNA. And what they do, they have to do the same thing that lytic and lysogenic viruses do. So what they will do is they'll take their RNA and they will use the cell to make it into DNA. And then the DNA makes more RNA and codes for proteins, the proteins for the virus, and it takes over the cell that way. It's called a retrovirus because it kind of goes backward. You start with the RNA and then you go to the DNA, whereas here you start with DNA and you make RNA, and then the RNA makes proteins, just like we've kind of already talked about. All right, so we're done with viruses. Our objectives for viruses was to describe the structure of a virus, explain how viruses infect cells, and define a retrovirus. All right, so now let's talk about disease. This is really very topical right now, and I'm not going to get specifically into coronavirus. I encourage you to use this information to look up what coronavirus does, how it infects a human host, where it came from. We think it actually came from bats. So I would encourage you to look at all of that, and we may have time to go into that. But for now, we're just going to talk about what is in your book. Now, there are two types of bacterial diseases. There is infection, where the actual bacteria is infecting some place in your body. An example of that would be like strep throat, okay? There are actual bacterial colonies living on your throat when you have strep throat. So that is a bacterial infection. Intoxication is where you have taken in, that would be like food poisoning, where you've taken in the toxins that the bacteria produced, and that made you sick, okay? So both of these things are what make you sick, how the bacteria makes you sick. Now there's two different ways of treating, because antibiotics don't really prevent. They may prevent disease, but they're not a great way to prevent disease because they also kill good bacteria inside of you that are doing great things. So antibiotics mainly are meant to treat bacterial infections. Okay, so they kill living bacteria. They're not going to help you treat the toxins that you may get from food poisoning or some sort of bacterial intoxication. So they treat infections. Vaccines, on the other hand, are a weakened or a killed pathogen that you inject into your body to make your body produce an immunity to the disease. Okay, I'm going to write that down. So what that means is if you are ever exposed to that disease, your body will already have the immunity for it. Okay, the last thing I want to do is just talk about the difference, some common diseases, bacterial versus viral. Bacterial, we would have strep throat, tuberculosis, Another one is diphtheria, which people don't usually get anymore. But those are just two. Viruses, you would see, the common cold is a virus. Okay, obviously COVID-19, which we're dealing with right now, is a coronavirus, named as such because it looks like a crown. Another virus would be the flu. Okay, influenza. So let's go back and just double check my objectives. Explain how bacterial infections cause disease. There's two ways, remember. Understand the difference between how we prevent viral and bacterial infections, and identify some common disease-causing bacteria and viruses. All right, if you have any questions, please feel free to email me or post them, or I don't know. This online learning thing is all new to us, and I hope you have a great day, and hopefully we'll see you soon.